Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report.
The study of microbes and their interactions is changing our understanding of biology, but only a small fraction of microbial species have been cultured. One of the challenges researchers have faced is the limits of existing tools used to study microbes. We spoke to Peter Christey, co-founder and CEO of GALT, about our emerging understanding of the microbiome, the need for a technological transformation of microbiome-based science, and how his company is hoping to fuel the emergence of new insights with its high-throughput technology to isolate and cultivate microbes. Peter, thanks for joining us.
Well, thank you. I appreciate the opportunity to chat.
We're going to talk about GALT, General Automation for Lab Technology, microbes, and the challenges with microbiome research. Perhaps we can begin with the opportunity and the recognition of not only the role microbes play in human health, but the potential to target and exploit them to address environmental, agricultural, and industrial applications as well. What's the excitement today to target and harness microbes across industries and application? What's the potential?
Well, I mean, that's a really interesting question and a very profound question, Danny. It's good to have the opportunity to talk about. Going back and I'll focus on human health and wellness first. Going back about 10 years, I think most people just viewed the microbes in the gut as being microbes in the gut, and they were potentially involved in helping digestion, and that was it. And a lot of the focus around human health and wellness was drugs and DNA sequencing and sequencing the human genome and such like. But in the last few years, there's been a huge amount of work that's recognized that the gut microbiome and the other microbes that live on the human body actually have a very profound impact on human health and wellness. And they do that in many, many different ways. And it's, well known now that there's a very large number of microbes on the human body. More than 50% of the cells in a human body are actually microbial in nature and not human in nature. And there's a very large number, multitudes of genes, microbial genes in the human body that far outnumber the human genes. So they're really an integral part of human biology, and this has been a realization that's just developed quite recently in science, I'd say in the last 5-10 years, and it's starting to really accelerate now. So not only is the microbiome been linked to many, many different disease states. Many of those disease states are not intuitive, that there would be a microbial connection to them. But also in terms of key biological processes, such as training the immune system, drug resistance or drug susceptibility of individuals that was previously primarily thought to be genetic in nature. Now we're finding the microbiome has a impact on drug response. And then even more recently, people have discovered that there's been a microbiome in different tumor types, and that can impact the growth of the tumor and the way it reacts to drugs. So what we're seeing in the human health and wellness arena is that this group of microbes that no one really took much notice of, 10 or 15 years ago, is now realized to be a critical and integral part of who we are and what we are as human beings. And there's a huge amount of interest now in understanding how these microbes do this. And so there's a lot of really, really great science to be done. So that's the human health and wellness field. The second aspect that you referred to is around agriculture. And again, in agriculture, there's been the first green revolution that started in the 50s. A lot of driven around genetic engineering and trait selection in plants and development of fertilizers and such like. And that really helped feed the world in terms of where we are now. But now the entire agricultural industry is focused on the issue of how do we feed, 9 or $10 billion on the planet or 9 or 10 billion people on the planet by the year 2050. And we need another green revolution. And the industry sees the microbiome of plants and how microbes can help plants become more resistant to pests and more productive from a growth perspective. That microbiome-related science is going to be a key aspect of how we can increase crop productivity to meet the needs of this expanding world that we live in. So I'm looking at it from a life science tools person and a scientist in this field, and I'm looking at this and saying the microbiome is so important in so many different fields, in human health, human wellness, in agriculture, and how we feed the planet, that it's just an incredibly important field of science that we need to do as much as we can to drive our understanding of the microbiome and how it works as fast as possible.
I think many people would argue that the microbiome revolution and world of biological projects has not moved as quickly as anticipated. What role has the available tools to study and decode microbes and their interrelated communities played in slowing progress?
I think the field is actually moving very fast. Remember, it's a very nascent field. It's only been really focused on in the last 5, maybe 10 years with the advent of next generation sequencing and the application of next generation sequencing to microbial communities, and really help understanding the complexity that exists in those communities. And that realization is actually quite recent. And there's been a huge, progress in the field that, in terms of not understanding the underlying mechanisms per se, but understanding that the microbiome is a critical factor, it's really important, and we have to understand it. So that's the stage that we're at now, and the field is very, very nascent. My concern is that, right, we have this realization, we have a viewpoint into the microbial communities, and we understand how important they are. And so what we need to do now is delve into the science and get a very detailed understanding of what's going on. And the tools that exist to do that today, to support microbiome research, as you go into a wet lab environment and do cell biology, they're not adequate to really answer the questions that we need to answer today.
Well, what is GALT and how did it come about?
This is a really interesting story. So GALT, the mission of GALT is to provide new tools into the microbiome research lab to enable microbiome researchers to answer the questions that they need answered to understand microbiome biology. That's what it's all about. So the way we started is a few years ago, I met up with someone who had come out of the ag bio industry, and he was very aware of this issue of cultivation being a problem. So what happens is that, you may take a sample of some type, say a soil sample, and from that soil sample you want to cultivate specific microbes of interest. And then when that sample goes into the cultivation lab, suddenly it hits technology that was invented 150 years ago. So the process of cultivating target microbes is really slow, really inefficient, and you may get your targets in a week, or you may get your targets in 2 years, or you may get only a small proportion of your targets. And that is a significant barrier to both the science of the microbiome as well as generating product candidates for downstream evaluation. And the reason it's so slow is people are in those laboratory environments. The standard is just using petri dishes and streaking hundreds and hundreds of petri dishes and picking colonies and then trying to generate candidates from that. And it's really slow. And this technology, as I say, the petri dish was invented in 1887. The Bunsen burner was invented in 1855. So that's the state of the art of technology in a microbiology research lab today. You compare that to a genomics lab today, where you have a lot of high-throughput analytical instrumentation that helps scientists do what they want, or if you compare it to a cell biology lab where you have flow cytometers, high-content screening and very fancy microscopes, none of those specific tools exist in a microbiology lab, and that's the reason things are so slow. So coming around, to how GALT was started, is that I connected with this individual, and we sat down and talked about it, and we realized that this was actually a very generic problem and very well recognized problem in microbiology, that microbes were just very difficult to cultivate from environmental samples. And there's a number out there that is in the literature that less than 1% of the microbial species on the planet have ever been cultivated in a laboratory. So there's a huge amount of richness and biodiversity that we just don't understand and can't access because these cultivation techniques are so archaic. So we talked to a lot of people and found out this was a very generic problem, both in academia and in industry, to a point that many people weren't even attempting to cultivate from samples because they said it's just too difficult and too troublesome. And at that stage of the game, there was a general acceptance that was just the way it is. Things are difficult to cultivate, and so we've got to find other means to get around that. And so Mike and I sat down. We said, no, we don't accept that. We believe that this is simply a problem and that this problem can be solved. So we started the company to do that. And then we went through and said, right, step by step, what do we need to do to address this problem of cultivation? Because we knew that the downstream impact of that would be very fundamental to the field of microbiome science and the development of microbiome-based products. And the positive impact of solving this problem would be absolutely huge.
We've had great advances in sequencing technology. Why is that not enough with regards to understanding the microbes? What are the limitations of sequencing in that regard?
Yeah, well, sequencing is just such an incredibly powerful tool, and it has catalyzed our understanding of the microbiome and how important it is. But it's not it's not 100% solution. It doesn't provide you everything you need. And I think the very core limitation of sequencing is that it's a see but not touch modality. So it allows you to get a snapshot of what is in a microbiome in any situation. So with sequencing, you can measure the microbiome of a healthy individual and compare that to an individual with a disease, and you can look at differences, and you can say, right, based on those differences, I have a hypothesis about what may be going on. Or you may look at a person who has a disease and then monitor that over time, and then look at the microbiome over different time points, and you can see how it changes. And again, based on that, you can generate hypotheses. If you want to reach in, and if you see a really interesting microbe or a species, and you think, I know what that species is doing, or I think I know what it's doing, you can't pull that species out, take it into a lab, and test your hypothesis and prove it. So next generation sequencing is great at understanding complexity, but generating hypotheses about mechanisms it's very good at. But in order to validate those hypotheses, you need to take isolates into a lab, into a controlled environment, and do testing on them to validate or not your hypothesis about how that microbiome is causing or impacting a particular disease state. And the analogy I draw is between cancer research and microbiome research, is that in cancer research, there's a really close relationship between genomic analysis and understanding what's going on genetically, or from a DNA perspective in this particular tumor sample or this particular cancer type, and the cell biology that occurs in the lab, where people are working with cancer cell lines. They're doing pathway analysis, they're doing knockouts, they're doing drug screens, they're doing a lot of cell biology in the lab that feeds from the genetic information, and then the cell biology insights then go back into the bioinformatics and then help people interpret the genetic data more effectively. So that loop works very well in cancer because you have the two elements working together. In microbiology and the microbiome research, the primary emphasis is currently just on next generation sequencing, because the tools in the cell biology sphere in microbiology are so antiquated that it's really hard to do those kind of experiments. So what we're doing as a company is targeting that and saying, how can we increase the capability in this wet lab or cell biology environment to increase the power of microbiome research so that the bioinformatics and the cell biology can talk together in a very synergistic manner.
You've developed the Prospector, which is a high-throughput microbial isolation and cultivation system. What exactly is it, and what does it do? How does this change what scientists are able to do?
So it does two or three things. So the first is that, one of the problems with the current tools in being used in microbiology labs is that they're very low throughput. So a typical petri dish, per device, if you're generating isolates from it, it generates maybe 50 to 100 isolates per device that you can pick off on that. So there's a necessity, and that's okay if you're working in a clinical environment or you're working with a single bacterium like Pseudomonas or Staphylococcus or Streptococcus from your throat or whatever. That works fine. If you're working in a complex ecosystem like a soil microbial ecosystem or human fecal ecosystem, these may contain many hundreds or thousands of different species that are all interacting with each other. So fundamentally, you need a far higher sampling rate or a far higher throughput capability, and so that your technology scales to the same level of complexity as your sample. So the first thing that the Prospector system does with its array-based cultivation approach is vastly increase the number of sampling points per device, so that you can start to generate isolates and generate insights into the microbiome that approaches the complexity of the microbiome itself. The second aspect is it greatly reduces the time and effort required to do cultivation experiments. We've mapped out the workflows of generating isolates using the prospector system versus the current technology. And there's about a 70 to 80% reduction in hands-on scientist time to generate those isolates. So this is really important in a lab environment where human resources are scarce and valuable, is that if you can reduce the amount of effort it's required to generate a lot of isolates in the lab for downstream screening, then you have a fundamental step change in your ability to actually analyze samples, pursue research strategies that include cultivation, and push your projects forward. The third key aspect of the system is that it's very, very compact. And I don't know whether you've ever been into a microbiology lab, Danny, but you usually walk into it and the benches are stacked high with piles and piles of Petri dishes or 96-well plates. And often you need a large number just to process one sample. So having a very compact, data-dense, sample-dense modality for cultivating frees up time and space for you to now, instead of processing one sample at a time because your technology is so cumbersome, you can now contemplate testing many, many samples in parallel and also testing many, many different conditions. So the speed and scope of your research goes up by an order of magnitude, and your programs and your insights develop so much faster. So that's the core of the system, is enabling just a really high throughput, highly efficient method of getting to the isolates that you want to study as quickly as possible in this lab environment.
What's the range of applications for this technology?
So the kind of things that people want to do with this technology, and it's kind of, the definition of an application, we could talk about, but, basically, the use cases, I think, that are really common. And cultivation is a really standard workflow in microbiology labs. So, every microbiology lab streaks things on plates and tries to cultivate. Every microbiome lab out there at some point is trying to cultivate isolates. So the core use case is just cultivation, making that faster, easier, and more productive, and getting a broader range of species out of your sample for you to study. So the use cases where that is people tend to use it. So the first one is building strain libraries. So, a lot of people are interested in building libraries of microbial strains that are derived from many samples, all over the country or all over the world, and producing a biobank of strains that they can then screen or have available to them as they pursue their studies. And then they say, hey, this study indicates that this bacteria may be interesting. I'll go to my strain library, pull that out and study it in more detail. Or else they're using it to mine for product candidates. So if I'm a large company and I have a strain collection that has 10,000 different variants of microbes from all over the world in it, and I'm looking for a particular feature, a microbial species that does something specific that I'm interested in, then I go to my strain collection and I screen that for the capability that I want. So what they want is a very rich and diverse biobank of samples that they can then mine for product candidates. Another use case is, slow-growing bacteria is a big one. So typically slow-growing bacteria are very difficult to isolate on Petri dishes because they tend to get overgrown quite rapidly by the fast growers on the array. So we're working with people now that are using our system to isolate fast growers in a far more, faster and cost-effective fashion. And the way the chip is engineered, it basically stops fast growers growing over the device. And so the slow growers have time to come up and be harvested. So that's another specific arena. And then I think a third use case, and this is kind of where we started, is that, if I'm an associate professor in an academic lab somewhere, and, I have a RA or a postdoc in my lab, and I'm working on a biological system, say I'm working on a disease state and I have patient samples, and then I do my various experiments and my sequencing analysis, and I find something really interesting, that a particular microbial species that, hey, I want to study that more, and understand, because this looks really interesting piece of science, then they use our system to go in and specifically isolate that microbe rapidly, so that they can take it out and then do whole genome sequencing on it. They can study its biochemistry, its physiology, and then test it in the disease system that they have, just to understand, is this bug doing something? Was my initial thought about that microbial species, was it correct? And now I've got the species in hand, and I can test it. And then, there's a whole new approach to, targeting an understanding of disease.
Is GALT unique in doing what it's doing, or are there competitors offering similar high-throughput microbial analysis?
No, this is a really interesting question. So the majority of folks that, out there are using Petri dishes and 96-well plates to cultivate bacteria. So the status quo is a competitor of ours. There is, kind of people out there that produce automation devices. So, there's colony pickers, for example, out there on the market. This is essentially automation of the status quo. So they take Petri dishes or they take 96-well plates, and what they've done is essentially build a huge robotic system just to process those things faster. These tend to be expensive. There's a lot of work moving the arrays around and such like, and whatever. But in terms of what's on the market, that's it out there. There have been some attempts in the literature and other folks kind of looking at, alternative ways of doing cultivation. But there's currently no alternatives for folks out there that, want to, change the way they do things.
What's the business model here? Are you simply in the business of selling equipment and reagents, or do you also do this as a service, or somehow engage in data collection or other applications?
So our business model is very clean, and it's well established, and it's selling instruments to the laboratory. The cultivation arrays that are used in conjunction with the system are consumables. So a customer will look at our system, say, yes, this can help me accelerate my research. They buy the instrument, they place it into the lab, and then as they cultivate different samples, over time, they use arrays and then buy more arrays from us. And so that's just the razor blade model. And this is similar, or exactly the same in concept to what companies like 10x Genomics do, what companies like Illumina do, kind of Applied Biosystems and Life Technologies on their instrumentation side. So it's a very, very well-established model. In terms of service, we currently don't have a service operation at this point. If people wanted us to process samples, we would, talk about that on a case-by-case basis, and we could do that if people were interested. But we don't have a firmly established service capability at this point.
We've seen a great drop in the price of sequencing. How much of a barrier is price to deploying your technology, and is there that similar type of opportunity to see advances in price and speed, or is this technology at a point where it's efficient enough for wide usage?
Let me see. So pricing. So a system that adds value significantly in the lab environment and is priced to be appropriately is this is going to be able to sell. So in terms of pricing or the way we think about our system, we don't want it to be such a expensive and complex system that only major research centers, kind of around the world can use it. We want the system to be something that any microbiome lab can have access to, bring into their lab and utilize, because the ultimate goal of what we're doing is to accelerate the whole field of microbiome science by by 1 or 2 decades, by providing tools to everyone who's interested in studying the microbiome and having access to that, so the whole field benefits from it and moves forward. Because as I started out at the beginning, this field is so critically important to everything that we do, and I see the role of our company as helping to accelerate that as much as possible. So the system, this is a brand new platform for life science. This didn't exist 4 years ago. We're just getting started in terms of the capabilities of the system, and we have a lot of exciting things to do with the system going forward. So over time that will develop. But currently, this in the state as it's in now, it has a huge amount of benefit to microbiome research labs, and we'll just build from that.
Peter Christey, CEO of GALT. Peter, thanks so much for your time today.
Good. Thank you very much, Danny. I appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.